And I want to make it bigger. All right, sorry, Ty. And okay. So let's get started. We ended chapter 2 and we ended it with the discussion on what are macromolecules, right? We started talking about bonds. So know those bonds, know how, just in general, what is a strong bond? Ionic and covalent bonds are strongest. And depending on where we're in a body, it could be a combination of those two. Whereas hydrogen bonds are not even real bonds. So they're the weakest of all the bonds that we've talked about. And we ended up talking about then macromolecules. So macromolecules are these really large combination of sugars and other atoms. Usually it's either oxygen, hydrogen, nitrogen, even sulfur, right? So these are the main macromolecules. So the ones that we're going to talk about, carbohydrates, fats, right? As well as proteins, as well as we'll talk about nucleic acids as well. Those are 4 main macromolecules. So let's start with the first one, carbohydrates. Now, carbohydrates have gotten a bad rap lately, right? So if you watch any TV shows, you'll realize that for some reason or another, sugars or monosaccharides have gotten a bad rap in terms of people gaining weight. Keep in mind that normally, when you take a blood sugar level, it is only glucose right here that we use as the instrumental fuel in our body. Now, do we ever really see glucose as a monosaccharide when we eat something? Not really, right? So when you eat real foods in life, you don't usually just take in glucose. The only time we give people glucose is if you're in the ER, you're not looking good, they want to give you some nutrients and they give you a little bit of dextrose. Dextrose is glucose, but that's the only time. But even then, that bag of dextrose they're giving you, it's kind of man-made. In nature, we never really see monosaccharides, right? Now, there are 3 of them, but we don't really see it. They're always found in combinations with itself, glucose and glucose, and we have a lot of glucose to make glycogen, to make starch, or you see it as a combination of 2 of these, fructose and glucose and galactose. Well, glucose and galactose combined is a disaccharide now, and now it is a disaccharide we call lactose, which is in our next slide. But in other words, even when we see it in nature, you never see each of these monosaccharides by itself. It's always in combination with lots of other isomers like glucose, and you have lots of other glucose, or you see the glucose with galactose combined, they form a disaccharide lactose, or you see glucose and fructose, then it forms a disaccharide sucrose, table sugar. That's a sugar we find in in grapes, in fruits. That's a combination. So why do we talk about monosaccharides then if we don't really see them in nature? Simple thing is that once we do ingest it, we break any of the disaccharides apart. So if you have sucrose, which I should have a picture of on the next slide. So if you have sucrose right here, sucrose is what we ingest. We will then have it go into our GI tract where we break it down, where we cleave this bond, this oxygen glycosidic bond. As we cleave it, we then break it apart. This part then becomes glucose. This part, a 5-member ring, fructose. We absorb each one independently. Now, the weird thing is this, there isn't that big of a difference between these, fructose and with a little glucose in it. In terms of structurally, I mean, it's really hard to see much of the difference. But when they're still combined with each other, they cannot bind to our receptors. They cannot bind to the glucose receptor because it's too big. It can't bind to a fructose receptor in our GI tract because there's an added glucose in it. So what happens is, just like with lactose, when there's an extra sugar, our proteins meant to absorb monosaccharides are so specific that when we have an extra monosaccharide, an extra sugar sticking out of it, it can't bind. So then it goes unaffected throughout your GI tract. Now, usually with sucrose, we don't have an issue with it, but lactose, we do. With lactose, we do have a issue. And the issue is simple. Some of you guys and Asians, I'm Asian, obviously, you can tell by my last name, all right? I was born in Indonesia. I didn't move to America until I was like 9 years old. So in our culture, in Indonesia, Chinese culture, we're pretty well known for being lactose intolerant, right? And what happens is, even as we were kids, you know, we love eating pizza and we'd have some other bad effects that a lot of American kids never did. And the reason for it is that lactose intolerance. And what happens with lactose intolerance is we are lacking the enzyme lactase. Lactase breaks apart lactose. What is lactose? Just this glucose and galactose combined with each other. The weird thing is, all we're doing is adding another sugar to it. That's all. So glucose added with galactose, that makes lactose. So you would think, right? How come we can't absorb both of them at the same time? Well, it's because our proteins are folded in such a specific way that when glucose and galactose are bound with each other, it can't go into the binding sites. It's like having a key almost looking identical to your house key. And you can't tell which is a key until you put it into the lock, right? As you put it into the lock, you realize it goes in, but it doesn't move. That's what's happening with our protein and lactose. Lactose can kind of get in there, but it can't bind completely. Because it can't bind complete with the protein does not allow that lactose to enter our cells. So then what happens? Lactose stays out of our cells. It stays as a disaccharide throughout a GI tract that should not be there. And now we have that bad combination, right? The bad combination of a solute, lactose, that should be absorbed, but isn't. And next thing we know is that, right, because you don't absorb it, water is going to be drawn towards it. Remember, water is drawn to solutes. And when water is drawn to the solutes, you have less water reabsorption in the GI tract, more water in the GI tract because you didn't absorb most of it. What happens when you have water in the GI tract? It's pretty bad, right? You have diarrhea, and now you have diarrhea going into the large intestines where your microbes are, our healthy gut bacterium are, and they're gonna get that lactose and they can break it apart. So then as it breaks apart the lactose, then we have gases that are produced. And now we have the kind of bad explosive diarrhea that you sometimes might have heard of. And it's all just due to the enzyme being lacking, and the enzyme is lactase. So again, we never find the monosaccharides as monosaccharides in nature. They're always found as either disaccharides, glucose and fructose making sucrose, or they're found as glucose and galactose making lactose. Now there's another one called maltose, but maltose is going to be glucose and glucose combined, but we don't really see it until we're going to digest starch. Let's talk about starch. Starch is a type of polysaccharide. Now the easiest thing to understand about polysaccharides is that there's three main forms. Now there's other subforms, but three main ones. And the ones that we see in our body is glycogen. Now, keep in mind, why do we want to store starch? Simple, right? Glycogen is our storage form of glucose. I kind of told you guys earlier that glucose, fructose, and galactose are the monosaccharides. We can absorb each one and introduce it into the bloodstream. But once all those forms of monosaccharides hit the liver then transforms everything into glucose. So if you take a look at all of these monosaccharides that we mentioned previously, they have the same chemical formula, C6H12O6. The only difference between this, and this, between the three monosaccharides, is the location of the carbon-oxygen double bonds and the location of the hydroxyl group sticking out. In other words, they have the same chemical formula. The difference is where we see those specific type of bonds. Now, because they're made of this basically the same material, C6H12O6, it can be converted. You can absorb fructose, but how many times have you heard about blood fructose count? We absorb it. Why don't we actually see it? Because once it goes in the liver, it gets turned into glucose. How about galactose? Same thing. We absorb it, but once it gets into the GI tract, into our blood vessels, we are then going to take it to the liver immediately. And then we are then eventually going to transform it into glucose. So again, eventually everything gets converted to glucose. You can absorb each one, but right, once you absorb it, it gets moved towards the liver and then converted to glucose. Now, because glucose is so important right here, when you have a high starch diet, what we're going to do is take any excess glucose. Now again, right, you're going to take a minimum amount and let it be used for the rest of the body. Your brain, your spinal cord, your nerves all require free glucose. So the free glucose that you just absorb, it's in your bloodstream. Well, immediately it gets used up by the brain, the spinal cord, and the nerves. Now, other organs in your body can use different sources of fuel. Once we have used up all that we need, right? So all the brain is used up. It's now in a good condition. It's full. Those cells are full. Now what we're going to do is usually we have excess amounts of glucose. So we need a way to store that glucose. How do we store that glucose? We turn it into glycogen. Glycogen is formed by animals, including us, and is basically glucose molecules bonded with another glucose molecule, bonded with another glucose molecule, all the way down. There's going to be thousands and thousands of glucose molecules that are bound together. And if you take a look, I want you to take a look at these bonds right here. This is called a glycosidic bond because it's going to bind, right, sugars, which are glycosides. And what we're going to have is this glycosidic bond faces downwards. Notice that? As it faces downwards, we call it an alpha bond. Our enzymes can break apart this alpha bond. So what we're going to see is not only is glycogen produced by the cells, our animal cells, but we also see this glycogen in a very similar role as starch. Starch in foods is glycogen, but only in plant form. In other words, our body can then break it apart, the starch we eat, and then once we break it apart, eventually, what are we left with? Just free glucose that can bind to our receptors, introduce into our bloodstream, go into the liver, and if you have enough for the body to use, any excess will be turned into glycogen. So we break it apart in the form of starch, then use whatever we need once we absorb it. Then any leftover, we reform into, right, human form of starch, glycogen. So the only difference between glycogen and starch in function is where we find it. In starch, we find it in potatoes, things like that, right? In humans, we don't have starch, we have glycogen. By the way, this is the reason why if you were an athlete in high school, you will always have a coach that tells you to eat lots of pasta 2 or 3 days before a meet, 2 or 3 days before a competition. Why? If you eat a lot of pasta, you're ingesting a lot of starch. Lots of starch means lots of these glucose. And what happens? You can absorb the glucose, break it down, absorb it, and then because you've eaten so much of the starch, you have so much excess glucose that it allows us to form glycogen inside our skeletal muscles. The problem is this: there's only 2 places where we store glycogen, and that is in the skeletal muscles throughout your body and in the liver. That's it. And there's only so much room for glycogen to store. If you eat starch beyond that, now you've filled up all your stores of glycogen because you ate so much of the starch, right? You have so much glucose in your body. Your body doesn't know what to do with it, so it stores it as glycogen first. But now all your storage of glycogen is then going to be full. What happens? The rest of that glucose gets converted to fat. So there's always that kind of balancing point, right? If you overeat pasta because you want to get ready for a meat, if you way overeat, any excess glucose doesn't get converted into even more glycogen, it gets converted to fat. In a second, I'll show you how that happens. Now, the weird thing is cellulose. Cellulose is found in vegetables. And the only difference between cellulose and starch, right? Remember, cellulose is glucose. Same thing, right? Long polymers of glucose. Just like starch, just like when we talk about glycogen. So why is it that we call cellulose fiber, right? The reason is this. Cellulose has glucose bonded with glucose, bonded with glucose, bonded with glucose, bonded with glucose, just like what we see in glycogen, just like what we see in starch. The big difference with glycogen is that the bond between the glucose molecules face upwards. Instead of this facing down, it faces up. Because it faces upwards, it's got a physical nature that doesn't allow it to bind with our proteins. Remember, the enzymes, in order to work, need to have a particular shape. As it has a particular shape, it has to be able to then react with the substrate, with the material it's supposed to work on. It's called a substrate, right? So it's supposed to act, and it's got that substrate, the glycogen right here, because its bond right here faces downwards, the enzyme can go in there and its binding site fits perfectly with this bond. And now it can cleave and cut apart this bond. With cellulose, even though it's still glucose and glucose, right, the bond faces upwards. And now there's a physical hindrance. And then that cellulose can't get to the binding site of our enzyme. Because it can't get to the binding site, it can't get digested and get absorbed. Cellulose then goes through our GI tract without being broken down, without being absorbed. Now, by chewing, you can make it smaller, right? But you still doesn't mean you're going to be able to absorb it into the cells. The best way I have to kind of describe this to you is simple. Have you ever eaten a steak dinner that's got all the proteins? It's got, all right, also mashed potatoes, things like that. When you go number 2 the next day, do you see steak and potatoes? Of course not, right? Why? It's because it's completely digested. How about when you have a salad? Or God forbid, right? How about if you eat corn? If you've ever had corn on the cob, I love corn on the cob, right? That corn has lots of cellulose in it. You can chew and chew and chew, but what you see on the other side when you go to the bathroom looks very similar to what you see when it comes in. Why? It's because it's got lots of cellulose that we can't break down. So it goes through our GI tract, basically whole, and it goes out the other direction, basically looking very similar to what it started with. That's cellulose. And the only reason we can't break it down is because the bonds, instead of facing down like we see here, faces upwards in a beta linkage. So other roles we'll talk about, energy, obviously, right? But we're gonna see that some of the carbohydrates gets turned into ribose and deoxyribose for DNA and RNA. And we'll talk about its role in terms of signaling and the ABO blood groups later on. Now let's talk about the other type of fats, lipids, other type of macromolecules, lipids. The big one being triglycerides. Triglycerides is our storage form, right? Excess triglycerides are added to adipose cells to increase energy storage. So when you overeat, even if you I mean, you've seen people who are vegetarians and they're still overweight, right? Why? It's because they're overindulging in either fried foods or they're overindulging in carbohydrates. So take a look at the structure here. Carbon, and then an oxygen and hydrogen. This is called a hydroxyl group. So when we talk about a triglyceride is a 3-carbon sugar called glycerol and 3 long fatty acids, right? When it combines, we call it triglyceride, and this is our storage form of energy, right? There's a limit to how much glycogen we can store. There's only so much room in the liver, so much room in the skeletal muscles. There's almost no limit to how many triglyceride molecules we can store. Keep in mind, our body likes to preserve energy. It expects us to have times where we're not eating, so then we will break down any stores of triglycerides when we're either fasting or, God forbid, starving. So our body likes to store triglycerides. And if you've ever seen My 600-Pound Life, you will see the limit is completely, right? We think that there's no way you can get that big. But my daughter and I watch a show together, and it's kind of crazy just to see these people that are 5 foot tall, 700 pounds, right? And they all have this huge amount of triglycerides in their adipose cells. Adipose cells are fat cells. Fat cells store triglycerides. The more triglycerides, the more and the larger the fat cells are. Right? Now, with triglycerides, how come and how do we get glucose from and turn it into triglycerides? So let's take a look at glucose over here. Right? If you take a look at the straight form of glucose right here, it's got carbons and hydroxyl groups everywhere. If you only take a look at this bottom part, this carbon 4, 5, and 6 of glucose, right? Take a look at it and then compare it to glycerol. You go back. And glycerol, it should be identical. In other words, when we have an overabundance of glucose in our body, it's easy to split it apart. So then we have 2 glycerols that we turn out of 1 glucose molecule. Then we have even an overabundance beyond that. We can convert it to fatty acids, and now we can form triglycerides, right? Because these sugars look very similar, it can be intraconverted, right? Glucose can get turned into triglycerides. Triglycerides can get converted eventually into a different form of glucose for our body to use. So they're easily interconverted. And our body likes to store as much nutrients as possible. So when you overeat, there's a limit to how much glycogen you can store. So you don't want to waste the rest of the glucose. What do you do then? You turn it into something else because you kind of filled up your glycogen stores. So what do you turn it into? Triglycerides. And triglycerides are our preferred storage form because there's almost no limit to how much we can store triglycerides. And the only purpose they have is, especially triglycerides, only purpose is energy storage. That's it. All right, lipids, fats, saturated versus unsaturated. I have seen so many studies that discusses saturated versus unsaturated fats, right? Now, I'd never tell people to just eat lard, right? Or eat just animal fats, kind of gross, right? But if you are eating a steak and there's a little bit of fat on the sides, how harmful is it that you eat it? Well, there's a little bit, usually it's not going to be that harmful, right? Keep in mind that saturated fats are what we see in animal fats, right? Now, if you don't do it too often, it's usually not a big deal, but you will see studies that shows you definitely should avoid it. And other studies that shows it's not a big deal, right? As long as you don't eat, steak and its fats every day. So it's always kind of that balancing point, right? It's up to you to choose. And there are studies that kind of show one direction versus the other. And even more kind of conflicting is that the studies are usually founded by the meat or the egg industry or by other kind of the almonds and the nut industry. All right. Again, the meat industry will try to get you to eat more saturated fats in animals, whereas the almonds and the nut industry will try to get you to say, they'll say saturated fats are bad, use unsaturated fats. Now, what is the difference? Because it sounds like there's a big difference between a saturated fat and an unsaturated fat, right? Structurally, the actual chemical, right? There isn't that big of a difference. This is a saturated fat, palmitic acid. If you take a look, what do we mean by saturation? Saturation means that all of these carbons are saturated with hydrogens. See that? Carbons can form 4 bonds, right? So if you have a middle carbon right here, it's got 2 bonds going to other carbons. And now it's got 2 other bonds going to hydrogen. So it's saturated in terms of the bonds. Not a huge difference, right? So here's an unsaturated fat. What happens? In an unsaturated fat, what happens is there's a carbon double bond in the middle here, meaning that there's no hydrogen on each one of these carbons in our double bond. Because there's no maximal number of hydrogens, it is unsaturated with hydrogens. That is the only difference in terms of structure. You think it'd be this huge difference, right? It's not, right? Because of that unsaturated nature right here. This linoleic acid is going to be more of a liquid in room temperature, where there's going to be more of a solid at room temperature. Now, the worst type of fats are the trans fats. The only good news is that they're basically going to be outlawed, and they've been kind of removed from restaurants. Nobody can really use these trans fats anymore. Right, other type of fats, phospholipids. And we'll talk about phospholipids in a few minutes as we talk about chapter 3, and we talk about the nature of our plasma membrane. Keep in mind, we have 40 to 50 trillion cells that makes us who we are. Each of those 40 to 50 trillion cells are going to be made out of these phospholipids that forms a bilayer. The phospholipid has an end over here. The end is going to be called the polar end, right? I'll answer that in a second. All right, so this nitrogenous end, that nitrogen has charge. Phosphorus has charge in it. So, right? And what happens is that all those charges right here makes it that it wants to react with other charged particle. So when we have a cell, we're going to have fluid and water on one side, fluid and water on the other side of the cell, right? That water is going to be a polar covalent bond. Because it's a polar covalent bond, it's got all the partial positive and negative charges that we talked about last week. So all the partial negative and positive charges likes to react with all the positive and negative charges here in the polar head region. Now, polar molecules like reacting with other polar molecules. This is polar because it's got charge. It's got nitrogen and phosphorus. And because water is on this side, water also has partial charges. What it means is that water will attract the head region of our phospholipid. The 2 tail regions coming off are hydrocarbons, carbon and hydrogen. Those are all formed by non-polar covalent bond. Because they're non-polar covalent bond, it doesn't like reacting with water that's polar. So it is what we call hydrophobic. It wants to move away from all of the water. So the head faces water, the water on the side, and then the tail is hydrophobic, phobia, right? And what happens is it faces away from water. Now, remember what I mentioned. There's water on the outside of the cell, and then later on we'll see there's water on the inside of the cell. Because there's water on both areas, we're going to see that there's a dual layer. It's not just one layer of these phospholipids. There'll be another layer on this side of it that we'll see in chapter 3. So these phospholipids forms an important structural component of all cell membranes. Now, what else do we see in terms of fats and lipids? Eicosanoids right here Eicosanoids derive from fatty acids. Eicosanoids are chemical signals, right? They include thromboxanes, leukotrienes, prostaglandins. They produce inflammation and pain signals. When you take aspirin, aspirin blocks the production of these. So then you don't have as much inflammation and pain. What else, right? Now, what else is that aspirin is pretty good, especially low dose aspirin. And if you can, tolerate it, even higher dose aspirin, right? Aspirin is a good blood thinner. So when you think of a blood thinner, I don't want you to think of it as like a paint thinner where you can just remove it, right? Blood thinners are going to block the production of these three that can cause us to trigger clotting effects. Now we'll talk more about clotting, chapter 19 next semester. But just for now, no, eicosanoids derive from fats. They produce inflammation and pain signals. Aspirin blocks it. Then this is the one that also important in terms of fats. Fat-soluble vitamins are decavitamins. Vitamins D, E, K, and A, nonpolar molecules essential for normal functioning of a cell. If you've ever seen vitamin D, vitamin E, right? Sometimes you see it at GNC or Elite Nutrition, or you go to Costco and you buy it. And what happens is if you take a look at it, is it a capsule or a tablet? A tablet is like the Tylenol, right? It's dry, white kind of looking pill. It's solid. That's what a tablet is. A capsule is kind of squishy. Now, what you'll notice is vitamin D, E, K, and A should always be found in like a squishy capsule. Why? Well, these are fat-soluble vitamins. In order to absorb the vitamins, you need to have some fat, right? And what we're going to see is if you ever have a vitamin E capsule, take a needle and puncture it. You'll notice that there's a little oil on the inside. The oil is what allows you to absorb the vitamins. If you have a tablet and it's all just a big solid tablet of vitamin D without oil, you will absorb maybe 5% of it, right? That's it. In order to absorb these decavitamins, you need oil or fat for their absorption. And that's why if you squish up the vitamin E or D, right, capsule, you'll notice that it's squishy, and you'll notice that if you puncture it, that oil is found on the inside. Now, vitamin D is also found as part of milk, right? Now, I see it from time to time where they tout something like skim milk with vitamin D. Keep in mind, skim milk has very little fat in it. Because it's got such small amounts of fat, they can put as much vitamin D as they want. You're not going to absorb as much as you should, right? So the only time you should see like vitamin D in milk that's helpful is if you're drinking whole milk. Whole milk has more fat that allows for the absorption of vitamin D in a much more efficient manner. The last one also, steroids. Take a look at cholesterol right here. Cholesterol, bile salts, estrogen, testosterone, all of these formed by cholesterol. So I won't say it this way. I'll just say cholesterol is the framework to form bile. So we take, oops, keep choking on typing. We take cholesterol and we modify it. If we take cholesterol and modify it, we can turn it into estrogen. We can turn it into testosterone. We can definitely turn it into bile. So all of these are going to be formed from cholesterol. Now take a look at estrogen and progesterone and testosterone. We know they're completely different, right? Testosterone makes males more masculine. Estrogen make females more feminine, right? So let's take a look at this. What's the difference? Take a look at this structure all the way through. And take a look at this one. They're almost identical at the top, aren't they? Oxygen, hydroxyl, methyl group. You have a 5-membered pentamer ring, right? You have a cyclic ring right here. Another cyclic ring, 6-carbon cyclic ring right here. They're almost identical. The only difference is in this last little bit, right? Why is this so important? It's because testosterone and estrogen can be intraconverted really easily. You've heard of people that are taking anabolic steroids. Anabolic steroids increase their testosterone levels to such a great extent that some of the testosterone gets converted to estrogen. You've heard of them developing breasts as a result. That's why, right? You've heard of women, right, taking high estrogen levels, and sometimes, especially if you have certain diseases like polycystic ovarian disease, PCOD, that disease or symptom can increase estrogen tremendously. And because of the estrogen levels increase, some of the estrogen gets converted to testosterone. The voice deepens. You'll have male pattern hair growth as a result as well. So all the bad things, right, of being a male. That's what sometimes happens with PCOD, PCOS. Why? Because those can be intraconverted. They're formed by the backbone, the framework of cholesterol. And if you don't get enough cholesterol through your diet, your body will form it itself because your body always requires estrogen and testosterone. And even males and females have a little bit of both. Now comes the fun part, proteins, right? And then we'll take a little break. Proteins are going to be formed by amino acids. There are 20 different types of amino acids. And here they're showing us some of the types, right? There's 20 different types. And an amino acid, if we take a look right here, has a group on one side called an amine. That's a nitrogenous group. And then an acid group, carboxylic acid group. This carbon with oxygen, double bond, and a hydroxyl group on the side, this is called a carboxyl group, right? Or carboxylic acid group. So why do we call it an amino acid? Simple. It's got amine on one side and an acid on the other. The big difference between the 20 amino acids is what happens in this carbon, right? This carbon is what we call the alpha carbon. That's a carbon that differentiates one amino acid from the next. So we get something as simple as glycine. This carbon, so all amino acids have this in yellow that are identical to each other. The big difference is what sticks out right here. We call it an R group or side chain. This right here is just a simple hydrogen. Because it's hydrogen sticking out, we call that glycine, simplest amino acid. Sometimes amino acids are huge, like this, right? So now instead of a hydrogen sticking out, you have a methyl group, CH2. Remember methane is CH4. So when you ever see CH2, CH3, right, that's what we call a methyl group. It just means one carbon with all hydrogens. And then here you see a phenyl ring, and then a OH. This makes that, do you see how big this ring is? You don't need to know the actual structures. Don't worry about that. I just want you to know that there's a big difference in these side chains, right? It could be something as easy as just a simple hydrogen sticking out of it. That makes it glycine. Or it can be huge, right? And make it tyrosine. By the way, tyrosine right here is going to be used to make thyroid hormone, right? Tyrosine is also going to be made, right, it's going to be used to make certain type of neurotransmitters. This is a very important type of amino acid. Again, we've only talked about 2, there's 20 of them. So here's another one, phenylalanine. The only difference between phenylalanine and tyrosine, OH group at the end right here. Tyrosine has it, phenylalanine doesn't, right? And then you have aspartic acid that can be here. There's a lot of different ones. Aspartic acid combined with phenylalanine forms NutraSweet. And since they're amino acids and not sugar, phenylalanine and aspartic acid forming NutraSweet are equal, doesn't have has very low calories. Amino acids by themselves is not calorie rich, not like, right, high fructose corn syrup. So here there's about 4 of them that we talked about. There's 20 different types, right? Some of them we can make by ourselves. Other ones are essential amino acids, like you guys might have heard of when you watch the movie, I don't know, Jurassic Park, they talk about lysine. Lysine is essential amino acid. You can only get it through diet, right? You can only get it through other meats. Now, every functional area of our body, enzymes are proteins, right? Receptors that allow our cells to work are made out of proteins. Proteins always has a function. Unlike fats, right? When we talk about triglycerides, triglycerides only function is for energy storage. Proteins has a function. You don't make a protein just to make it, because it takes a lot of energy to make a protein. What do we mean? Well, in order to make a protein, we got to somehow bond two amino acids together, and then bond another amino acid to that, and then another amino acid. Each time you add an amino acid, it takes ATP or energy. So you don't just waste ATP, right? It takes a lot of energy to make it. And if you want to break it apart, well, you want to break it apart for a reason. And you don't make that, our body hates to waste energy. So in terms of proteins, they're made out of amino acids. And those amino acids, it can be thousands of amino acids, are bound together by a carbon-nitrogen bond that they call a peptide bond. Sometimes small proteins are called polypeptides because of it. But everything you think about in terms of hormones, there's some kind of proteins, right? Thyroid hormone, right? Kind of protein. Insulin, glucagon, type of polypeptide, small proteins. So these are all the functions of proteins, and I'm not gonna go through them, right? Regulation, transport, contraction, structure, there's a lot of them. They all have different functions, and we'll talk about some of the functions in chapter 3. Now, how do they gain their function? So what happens is this: when a protein is made, a protein, in order to make it, we need to find out which of the 20 amino acids goes where. All right, you need like an ingredients list. That's what the primary structure is. I want you to know these structures for the quiz. So the primary structure is a sequence of which amino acid goes where. We have 20 amino acids. Where does methionine go? Where does glycine go? So what we're going to see is your DNA is going to tell us how to make the protein. The DNA will tell us which amino acids goes where to make that protein. So in order to make the protein, though, we need that list of amino acid number 1 right here. Usually amino acid number 1 is your methionine. Then what's number 2? Then what's number 3? What's number 4? What's number 5? So the primary structure lists which amino acids goes in which sequence. It's a sequence of amino acids, in other words. Right? Again, in order for a protein to work, it needs to have a shape. If we don't have the shape, it doesn't work. Right? Now, there are certain diseases we'll talk about where the shape starts to mutate later on after we make it. And those are usually deadly diseases. But in order to make it with a particular shape, we need these protein structures to occur, and it causes bending of our amino acid chain into the protein structure. Now, the primary structure, sequence of amino acids. Then what happens? As we have the amino acids start to stack up and build and get longer, some of the amino acids start forming hydrogen bonds. So here, this amino acid, in terms of sequence, right, is pretty far away from this one. So these two, this one and this one, they're pretty far away in terms of sequence. There's about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, about 12 amino acids in between this and this in sequence. But what happens is, because the sequence is so long, it starts to bend. As it starts to bend, these two amino acid chains, or these two amino acids as part of the chain, starts to hydrogen bond to each other. So does this one. So do these two. So do these guys. right? As they bond to each other, they form hydrogen bonds, and gives it what we call a secondary structure here. That secondary structure looks like a beta pleated sheet, right? So these are small localized bends, small localized structures found in a much larger protein. So in a large protein like this, you will have areas that will have beta pleated sheets. You might even have an area over here that has beta pleated sheets, right? Now, that's not the only secondary structure. Other secondary structures, there's two of them that's important. Now, this one right here, because of how close they are to each other, right, it forms a ribbon shape in that area. That ribbon shape is called an alpha helical structure. Now, some of you might have heard of DNA being double helical structure, right? Here, it's only one helix. And this ribbon-like structure forms due to hydrogen bonding between adjacent amino acids. So this amino acid starts to hydrogen bond with this one. Then this one starts to bind with this one, right? When it does that, they form the beta pleated, the alpha helical structure. And both are due to hydrogen bonding. One, beta pleated sheet, hydrogen bond between amino acids, pretty far apart in terms of sequence, but close together in terms of space. Here, the sequences are very close and they form alpha helical structure due to hydrogen bonds between, right, almost adjacent amino acids. In a large structure, you're going to have a lot of different regions of beta pleated sheets, a lot of different regions of alpha helix. Right? Now, that's a secondary structure. The most important structure is this third one right here, called the tertiary structure. This one, we kind of take all those little ribbons that we see, the alpha helical structure that are localized. Here's another alpha helical structure. Here's a beta pleated sheet structure. Here's the alpha helical structure. Notice how many structures there are here, right? These are all localized in a specific area. Here's the alpha helical, beta pleated sheet here. So we have lots of these secondary structures because our amino acid chain is so long. They start to interact with each other. And what happens is that when they, right, start to bend and form the secondary structure, because of the presence of different other proteins that help bend this much larger protein, what we're going to have is make big folds right here. Whereas the alpha helix and beta pleated sheets are localized, small folds in a specific region of the protein. Well, the tertiary structure is folding it all together to form the actual binding site of the protein. So you're taking this part right here, and folding it together, right? Now, I use the analogy of a paper airplane. If you've made a paper airplane before, and this worked better if I can show you on like a video. That's why sometimes I use the in-class video, right? What do I mean by this is a paper airplane, right? In a paper airplane, can you take a piece of paper, fold it in half right away and expect it to fly? Of course not, right? That's a big fold, right? You can fold it in half. Well, in order to make it fly, you need to have smaller folds, right? So what do? You fold it in half, and then you make little tiny other folds on each side. That's a secondary structure, the small folds you're putting in there. And once you have all the small folds ready to go, then you fold the paper in half to make the actual paper airplane. Now, if you've ever seen the directions on making a paper airplane, those small little intricate folds at the very beginning are incredibly important, right? It's only once you got those small folds at the very beginning correct, then you can fold everything in half to make the airplane structure itself, right? That's what we're doing here. We're having small folds, the secondary and tertiary, the secondary structures the beta-pleated sheets, the alpha helical structures. That's our small folds. But once those small folds are all correct, then we can fold the paper in half, form the wings, all right, form the airframe in the bottom, and now it can fly. Forming everything in half, that's a tertiary structure. You're causing big folds, right? Causing areas that are very far apart from each other to fold next to each other. So then we can form the active binding site. Without any of these folds, the protein will mutate and will be non-functional. Now, the big folds to make the actual protein functional is called a tertiary structure. It can be caused by disulfide bridges. It can be caused by hydrophilic, hydrophobic interactions, right? Remember, you're not making proteins in a vacuum, right? You have liquid on the inside of the cell where we're making a protein. So it'd be water everywhere. And now you have to fold everything in half, all right? Water is going to cause some of the amino acids that are scared of water, that are hydrophobic, to be turned inwards. Some of the amino acids that are charged will go on the outside. So then it can react with water and get stabilized. So there are a lot of these big interactions that allows us to form these tertiary structure that allows us to then finish the protein, giving the protein the proper binding site, right? And again, without the proper binding site, a protein is useless. Now, there are larger proteins where we have one protein, add it with another, add it with another, add it with another. We call this a quaternary structure. And instead of, right, one protein just being completely functional on its own, you add it with a second protein and a third and a fourth protein. Once you add all those proteins together right here, take a look, there's 4 of these, right? Once you add it together, they stabilize each other. They make it more functional and efficient. We call that the quaternary structure. Quaternary structure is an interaction between different proteins, different protein subunits that interact so then they can stabilize each other. Hemoglobin, right? Myoglobin are examples of a protein that our main purpose is carry oxygen, but each of the protein in hemoglobin is actually 4 different subunits working together. This is like the Transformers that you saw, right? When you were a child, or if you have kids right now, I think they still have Transformers, where you have small little robots that combine to form a bigger robot, right? That's what the quaternary structure is. The small little robots are the small little amino acid chains. They combine with each other to form a much larger, much more stable protein. Perfect. Now, all right, we can denature a protein, cause a protein to unfold, right? Now, when do we see this? We see this all the time. If you've ever cooked an egg, notice what happens, not with the egg yolk, but with the egg white. The egg white, right, when you cook it, the egg white, when it first starts, looks pretty, transparent, right? It looks see-through. But once you start cooking it, what happens to the egg white? It turns white and fluffy, right? So what happens then? Well, what we're doing is by adding a lot of heat to that egg white, the egg whites have a lot of proteins in there. The egg white then will cause, that heat will cause the proteins in the egg white to lose its fold. Once it loses its fold, that is the denaturing process. Now, that happens to us too, right? So you've heard of people dying because of fevers, having, because of really high fevers, having seizures as a result. Well, what happens when we have high fevers? When we have high fevers, we're increasing our body temperature. And in the brain, we will start losing folding of the brain. So this just shows you a different way of looking at the slides, different way of kind of discussing the primary, secondary, and tertiary structures and quaternary structures. I put this on here. It's kind of redundant, but some people like to see it in a different way, right? So I just put that up there. right? Protein denaturation, we see it in high fevers due to infections. Now, some of you might have heard of mad cow's disease, right? Chronic wasting disease in deer. We have a human form of it called Creutzfeldt-Jakob disease, right? Sometimes called Kuru, because they first saw it in people in New Guinea in the 1960s and 70s, especially this form, the Kuru. And what they would do is they would eat. I know you've heard of people in those areas that were cannibals, right? Well, what they would do is they didn't eat people that they didn't respect. That sounds gross. But sometimes when an elder would die of old age or some kind of sickness, right, the people will ingest part of that elder's body. And usually in those areas, right, if you eat like the brain, they think it'll give you more brain power, right? So they would eat the brain of the elder. I know, it's gross. As they ate the brain, well, what happens is they started having the mad cows like disease nature, where they have this, it started out having a movement disorder. And then it's a progressive disease where it's like Alzheimer's as well. There's dementia that goes with it, right? And then eventually, after a few months, they just crash and die, right? Well, what they found out is that by eating the actual brain of the elder, there's a small infectious protein, not a virus, not a bacterium, even smaller than that, an infectious protein, a protein, right? That is infectious. And since they ate it, they will now receive that protein. It goes into the brain, and the protein we call a prion, right? That infectious protein will then go into the neurons and causes other proteins to lose its shape. And now all the neurons have lost the shape of the proteins. The neurons can't function. As the neurons doesn't function, at first you get the movement disorder. Then with further neuron deterioration, you start losing memory. And then eventually you stop breathing because you no longer have the brain capacity, right? Death occurs pretty easily, pretty readily because of it, right? Now, with all enzymes are a type of protein, right? Keep in mind that the main purpose of an enzyme or a protein is to cause some kind of effect. Usually you have a binding site. In an enzyme, here you have two binding sites. Molecule A binds here, molecule B binds here, and then you fuse them together right here. But the molecules have to fit on the binding site. Right. So just review the enzymes. The last thing we're going to talk about before our break is nucleic acids. Now we will talk more about nucleic acids in chapter 3. Whoops. In chapter 3. Nucleic acids, there's two forms: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA is double-stranded. DNA is what we found, or what's found inside our cells in the nucleus, is what we give away to our kids. You guys have heard that we have 46 chromosomes, all right? Each chromosome is a double-strand of DNA. We have 46 double strands of DNA. The difference between a chromosome and a double strand of DNA, really no difference, right? DNA is going to be a chromosome. The big difference is usually proteins are going to be wound on the chromosome to make it more solid, so that it doesn't take up as much room in the nucleus. So the only difference between DNA and chromosomes is DNA is unwound without the proteins, right? Whereas chromosomes are the DNA itself, but wound on a protein. Kind of like what happens if you have, lots of thread and you wind it into a ball. It makes it more solid, takes up less room, right? So DNA and RNA are composed of a sugar. In DNA is deoxyribose, so the sugar is deoxyribose. In RNA, the sugar is ribose, ribonucleic acid, ribose right here. Now, with DNA, you have a sugar and then a phosphate group. This phosphate group is the attachment point to another nucleotide or another, molecule of DNA, right? So in terms of DNA, it is formed by a phosphate group that attaches to the next one, a sugar, deoxyribonucleic acid, and then one of four bases, right? It can either be cytosine, guanine, thymine, or adenine. You have either one of four of these bases. With these bases right here, right, we're going to see these bases, thymine and adenine, guanine, or cytosine, ATGC. There's always, these two are always going to be found together. These two are always found together, right? We'll talk about why coming up, right? So these are the four bases in DNA that differentiates one nucleotide from the other. Then you have the sugar, deoxyribose, and then a phosphate group attached to it. The phosphate group attachment right here allows us to add another DNA nucleotide to it, and then another in the bottom here as well, right? So these are the attachment points. This is the sugar. This is the base that makes it different than the other one. DNA is almost always double-stranded. RNA is always single-stranded. Now, some other difference between RNA and DNA. RNA, the sugar is different. The strands are different. And in RNA, there's no thymine. Instead, you have a different base called uracil. Now, how come that did not show up like this? All right, let's take a look right here, right? I'm going to see what happened to my slide right here, right? When you take a look at this slide, right, it doesn't show it, but what I want you to do is kind of look it up right here. And what we're going to talk about now is what we call the law of complementary base pairing. That means in DNA, if we know this nucleotide has thymine in it, then the other strand should have adenine on it. So pink, they put as thymine. Blue is adenine. Pink and blue should always be together, right? Cytosine is yellow, guanine is green. Kind of makes sense, green and guanine, right? So you have thymine right here. Thymine is always with adenine, AT. Guanine, always with cytosine. So if you know one side of the DNA, you know the other side, right? This is what we call the law of complementary base pairing, right? So here's our one side of DNA, one strand on this side. Here's the other strand over here. Now, keep in mind, this is color-coded, right? Even though it doesn't have the base on here. This is going to be thymine. Thymine has a single ring, right? Because it's got a single ring, on the other side, you need something that's a dual ring. Now, if it's a dual ring, it can either be guanine and green, right? Or it can be, right? Or it can be the other one right here, right? Cytosine, right? Let me see. It's actually adenine. Sorry about that. Adenine's in blue, right? So it can be one of these two. Why is adenine with thymine? Or thymine with adenine? Why is adenine and thymine always together? The reason why is simple. You maximize the hydrogen bonds. When thymine is on one side and adenine is on the other side, you have two of the hydrogen bonds stabilizing that area. How come guanine and cytosine are together? Also, right, to maximize the hydrogen bonds. The hydrogen bonds are the only thing keeping our DNA molecules together. If we rip apart the hydrogen bonds, our DNA molecules will spread apart and it'll be mutated, right? It'll be no longer functional. So the only thing keeping our DNA and our chromosomes together are the hydrogen bond interactions between thymine over here in red and adenine in blue, between, right, guanine in green and cytosine, A, T, G, C. So if we know this area, right, has 4 bases and it's 25% adenine. All right. We should also know it has 25% thymine. Combined, that's 50%. Then these other two bases, guanine and cytosine, will be also 50%. And they should be found in equal numbers, 25%, 25%. In other words, by knowing one base in a particular region, if you know the number of that one base, you should know the number of all four of the bases because of complementary base pairing, right? Again, because of this complementary base pairing right here, and I do want you to know this for the test, for the quiz, all right? If we know that in a region of DNA, there's 15% of that region has adenine. How much cytosine do we have? All right, I'm not asking for thymine, I'm asking cytosine, right? It's a little bit more complex. If you have 15% adenine, adenine and thymine are found in identical quantities because they react with each other. So if you have 15% adenine, you should have 15% thymine. That makes 30%. The other bases, cytosine and guanine, will make up the other 70%. Since they're found in equal numbers, you should have 35% guanine, 35% cytosine. Again, by knowing the number or percent of one base, you should know all of them just by this law of complementary base pairing. Right. That's it for this part. All right, actually with ATP, understand for now that ATP is our storage form of energy. All energy requiring chemical reactions, all build-up anabolic reactions require energy. When they require energy, they require ATP. ATP is energy, right? And the energy is found in the bonds between the phosphate groups. You see the squiggly lines right here? That's what we call a high energy phosphate bond. When you rip it apart, electrons are released. When electrons are released, electrons give out the energy required for a reaction to occur. Perfect.